KRAS has been looked at. It's a mutation that's found in many tumors. It does seem to be generally correlated also with radiosensitivity. There's some data from the Harvard group where they looked at proton radiation for liver tumors. And everybody there had always thought that size was an issue, but they were, with protons, were able to get high dose into large tumors. And they found that their tumor control rates were really not correlated by size at all. But there they found KRAS mutations and P53 mutations to be correlated with risk for failure. It's not been well studied in lung SBRT. There are a couple smaller series that there's some suggestion that KRAS may be correlated with local failure also. For ASTRO this year, right, we looked at that within our lung SBRT data set, right, and we really found no correlation at all between KRAS mutation and local control. Again, it's hard to find a data set that's big enough, but that was by far the most common mutation in our non-small cell lung cancer patients. About 40% of them had KRAS mutations. Again, I think we're in the lung setting. What's different is we're typically treating small tumors and we're typically treating them to high dose. So it at least feels to us, reasonably comfortable that KRAS status does not drive local failure in these small lung tumors, provided that you're treating to adequate dose to begin with. And that may be the difference, right? In the liver, you're not always able to treat to adequate dose with big tumors because you're limited by normal tissue toxicity. In the lung, most of the regimens we would use seem to be adequate. Last year, we actually looked at our liver tumor experience and lung tumor experience, not in non-small cell lung cancer, but metastases patients, and at least found something similar, right? For the relatively high dose regimens we tend to use, KRAS didn't seem to drive local failure. I guess the disclaimer is these are still pretty small data sets, right? There's not huge numbers of KRAS patients in them. So it's, could we be missing an effect that's not a huge effect? Certainly possible, right? But at least it's, in the Harvard study, it was only 50% local control for the KRAS positive patients with P53 mutations. Certainly, at least we feel comfortable that in the lung, the difference is not such a huge difference. If there is one, that we're not able to pick up because of our numbers, it's a relatively smaller difference when you're using high dose radiation. And the other part of it, I guess, right, we also looked at PD-1 status. That did not seem to correlate with local control. Interestingly, when we looked at overall survival, the p-values were all non-significant, but it does seem like, there's some layering of survival, where in fact, the PD-1 100%, which are patients that generally respond well to immune therapy, right? So medical oncologists get excited when they see that because we think these immune therapy drugs would work really well. There was a trend towards lower survival for the PD-1 50 to 100% patients, which again, I think we need a bigger data set, right? That's not anything definitive at this point. It's we'd love to combine that data with other institutions and see. But if that were true in a bigger data set, these immune therapy drugs that are out there, work best in patients with high PD-L1 counts, and SBRT patients, while they can't tolerate chemotherapy or major medical interventions, they can typically tolerate immune therapy. So if that were, I think there's not a lot of big data sets looking at PD-L1 status for stage 1 disease, or it's something we think of mostly for stage 4. But if that were to bear out in a bigger data set, it would be a great opportunity to think about using some of these drugs to improve outcomes in those patients along with radiation.